Great, we're winding down. So now let's get on to the bacterial cells. We have a whole section that has bacterial, animal, fungus, plant, all together in the microbiology, and you'll be able to go through that course shortly. So the bacterial cells, when you look at them, one thing that's very different about bacteria cells than eukaryotes are eukaryotes, you see the nucleus in the middle. It's one of those identifying characteristics. With a bacteria cell, it's a prokaryote. Pro means before, so it was before the karyote or the nut. So the nucleus is referred to as the nut, the karyote. Pro means before. Eu means true, or the standard, or the normal. So a eukaryote has a normal little nut in the side of it. Looks like a little kernel in there. A prokaryote means that this thing developed before the eukaryotes came along. So if you look at the structures over here, whoops. Here you can see the prokaryote. There's no nucleus. There is DNA inside of it, but it's all kind of wound into little rings, where over here with the eukaryote, you can see that solid ball of nucleus, really dense. It has a nuclear envelope. It's actually packed densely in there, and that's why you see that dot in the middle. So prokaryotes also don't have the organelles that the eukaryotes have too. The way they divide is different. So they divide by binary fission, which we talked about before, where you have that one cell. It replicates everything on the inside. It starts pinching, and then boop, it's into two. Where eukaryotes will typically come together and develop by meiosis, where you have two parent cells that come together, share their DNA, and then they replicate and have a baby. So prokaryotes, one characteristic you want to watch for, which also means that they're unicellular. They only have one cell. You don't see them stacked like you see in human tissues or plant tissues. The morphology actually describes the shapes. So there are really three typical shapes the bacteria come in. They come in causi, so a ball shaped. They come in spirella, which is spherical or like a spear. And they come in bacillus, which is rod shaped, like long. And then you can actually tack pieces together. So if you know those three words, they tell you the shape. Then you can put things like strepto, which means a strip of. So if I have a streptococcus, I have what shape? I have the sphere shape. So I have a strip of these spheres, so it's just a bunch of balls that are stuck together, like a long chain of like a pearl necklace. But if I have a streptobacillus, then I have a what? I have a long section of rod shapes. So almost like if you've seen the older shows where they have the sausages twisted together, where it's one sausage connected to another and another and another, that's what that would look like. There are some other shapes too, but those are really the three primary shapes. So morphology describes the shape of the cells. And when you look at under the microscope and you see them wiggling around or moving around, if you can see close enough, you can actually start naming it. So are they alone? Do they have chains, strips? Do they come in bunches or bundles? Right? And we're going to try to find some of these actually on you, some bacteria on you. One important thing to know about bacteria is that it can form something called a biofilm. And biofilms are like little cities of bacteria is the way you could think of it. So a biofilm is when something starts developing into a sheet across the surface and it's just packed with lots and lots of bacteria. It's the biofilm, or the bacteria that made that biofilm. It's almost like it made this little city, and it's really strong. It's hard to kill when it's harder to kill when it's in a city. Places you can find biofilms are, well, in bad places you can find them. If you're in medicine, when people have an IV in their arm for too long, if a bacteria gets on that IV, it can actually develop a biofilm, which means it's really hard to get rid of. And because there's a lot of bacteria, it can be dangerous for the patient. Your teeth, actually, when you have plaques or tartar buildup, that causes a biofilm because bacteria can hide inside of that and they can get more and more numbers, which means they're more and more aggressive about destroying your teeth. And that's why it's important to get rid of biofilms in your teeth. Other than that, also it can increase the risk of sickness. So if you can think of other places like well, I cleaned out my refrigerator and I pulled out the drawers and there was some filmy stuff on the bottom, and I'm sure that was a biofilm. So it was a drip from a jar of something, but there had to be bacteria growing in it. I think of my shower curtain, that's mostly minerals, but anything that has like a rough surface, bacteria can hide and grow in there. So it can start turning colors. You might get fungus that grows in there. Bacteria, the fungus usually will change the color of it too, so it looks all nasty. Well, let's see if we can find a biofilm on you. What you're going to need is a glass slide, just like this one, cover slip, just like that one, and you'll need a new toothpick, just like that. And this is always gross. I love showing people how to do these things and test yourself, but at the same time it's kind of gross scraping your own tartar. So what you're going to do, take your glass slide, you're going to try and scrape. And be very careful because it's sharp, but just above the gum line, not actually touching your gums, the pink part in your mouth, but on your teeth, and you're going to see if you can scrape anything off right along that gum line, or the front of the tooth, or inside the tooth, or whatever, but make sure you're only scratching the teeth. You can also test this with your toothbrush, so you can take your toothbrush and put in a little bit of water and just kind of rub the water of the toothbrush across the surface of the slide and use it that way too, but let's see if we can get something off. So I'm actually using it kind of like you'd use a toothpick after a meal. Didn't very much off. Flossing really paid off today, I guess. I got a little bit there, right? So what we're going to do on the next page in your manual, let's see, we're going to make a smear slide. So with the smear slide, I said smear like it's cream cheese on a bagel, right? I'm going to put a little bit of that right in the center. You can really tell, so I'll put this here. After scraping most of my teeth, I got a little bit. I don't know if you can see very clearly. Right here above where my finger's at. Does that help? I just scraped it off the toothpaste, or the toothpick. All right. What I'm going to do is I'm going to take my methylene blue, right here. I'm not going to use the same toothpick. I don't want to contaminate my methylene blue. Take a little bit. I'm going to put the methylene blue right here next to it. Drop of water on here. Just one drop. I don't want to go crazy. Boom. I'm going to take the toothpick and I'm going to smash up that little piece of plaque, tartar. Did I get it? Keep smashing. I'm going to put my finger over so you can see. I just got my fingerprints on the bottom, so I'll wipe that off too. Little piece. Pretty aggressive. Okay, I feel like you use a little bit more blue. I'm not going to stick this back in. I actually have another one that I didn't contaminate anything with. Drop. I'll wipe that off. Because of course I'm going to get some of that tartar onto this, so these two toothpicks are garbage. So I have my garbage can over there in the other part of the room. And now I'm going to take my cover slip, and hopefully you can see this. I'm actually going to drag, I'm not dropping, just dragging back, turn it around, go the other way, dragging it, I'm just smearing it. Smear, smear. If I had more tartar, I could have just drugged the bare tartar across and then stained it. But I didn't have a lot really to work with here. And if it's a really thin material, you can push back the other way. Drag it back, push it back. And I can see I have little tiny fragments to the side, so I'm going to push those in. Alright, now, one thing I could do, and I would not do this unless you had adult supervision, but you could actually take that, hold it with your tweezers, or put it in something safe like the edge of cork, or with a hot holder, and put a lighter underneath it or a match and kind of heat that up, and it'll dry out your biofilm. And then we're going to take it, slide it underneath. Just get that out of the way. Thumbs bring up the spring clips, sliding it back, turning on my underlight coming up, and let's see what we have here. There we go. Let's see. Get a closer look at it. The camera's still trying to auto zoom in and out. Alright, that scratches. There we go. Cool. I just happened, there's a big bubble. I just happened to put it right over a cluster. You can see some didn't get completely stained. But that's okay. Let's see if I can find some individual cells. There's some individuals. Yeah, it's a small cluster. Let's see if we can turn that brightness up a little bit. Nope, that's all I got. Might be about time to change my batteries. Alright. So we're pretty clear. Let's go ahead and flip our rotating nose piece. Alright, I'll come back here while I'm doing this. Turn the revolving nose piece right here. Now we're on the 10x, low power. Oh, big bubble. Get a little bit bigger picture of that. Oops. There we go. Now I'm just going in and out, and I'm looking for anything that looks like it's moving. Not really. Okay. So we're at 10. Objective, 10 ocular, that's 100 times total magnification. I'm flipping it over for the next magnification. Nothing moving. So right now I'm at the 40x objective, 10x ocular. I'll look around, see if I can find that cluster again. There's some blue, see what I can find. There we go, there are some cells. Not seeing a lot of movement. So I'll look around. Right now I'm at the right perspective. So I should see everything at that level. Is that moving? No. Of those cells. So let's keep looking around, see if I can find that big cluster. See if I can find any bacteria hiding in there. Oops, individual cell. Ooh. All those little tiny specks, I'm not seeing them super clearly, but I'm expecting that to be bacteria. There's a little something moving up there. I don't think I can get it clear. I saw something moving along the edge. Let's see if I can make this look a little bit better. Well, let's move on to the next cluster, see if we can find it. Ooh, there's the edge of the glass, and ooh, it's a nice group. Oops, too high, too low. And I lost it. Oh, oh, oh. Snap a picture. Let's look around. So I see some cells. And unfortunately, as soon as I make this, as soon as I switch my ocular to the 20x, we won't be able to use my camera. Wow, that looks like it actually has a bunch of bacteria on the surface. Okay, let me try this again. All right, I'm going to switch to the 20. Yep, so here's my 20x. I wish it were just a little bit longer to give my camera a little more stability. Yeah, not happening. Okay. Well, you can switch to your 20x, pop it in, go down to your scanning, find your cells again, which I'm going to have a hard time doing if I keep using my eyepiece. Find your cells again, focus, and do the math. So if this says 4 and this is 20, you have 80 times magnification. Flip this up. This says 10 and this is 20, so I have 200 times magnification. Clear, make it clear, and then finally go up here, which this says 40 and this says 20, so I'm at 800 times magnification. So a lot better. And usually about 800 to 1,000 times magnification, you can see bacteria cells pretty clearly. So I wish that you could take a picture for me, but you'll have the same issue with using the camera too to take the picture, is that you'll be able to see it with your naked eye, but you won't be able to get the image off. So that's making a smear slide. So there are a couple new techniques that we haven't used before. There's a lot about information about cells. We talked about prokaryotes like animal cells, plant cells. We talked about what makes a plant cell different than an animal cell. We talked about how they're both eukaryotes, but a plant cell has chloroplasts because it's an autotroph and can make its own energy out of sunlight, so that's what gives them the green color. We talked about a cell wall. So a plant has a cell membrane like an animal does, but it also has that firm cell wall, and that's made of cellulose. And that's actually the fiber when you eat a healthy meal that's full of good fiber. It's the cellulose from plants that you're actually eating. So there are a couple characteristics that make a plant cell different than an animal cell, and then a lot of characteristics that make a bacteria cell different than animals and plants. So bacteria is a prokaryote, where the other two are eukaryotes. Prokaryote meant before the what? Before the nucleus. So karyote means nut, but it's before the nucleus because that looks like a little nut in the center of the cell. The DNA, they both have DNA, but in a eukaryote it's in the nucleus. In a prokaryote it doesn't have a nucleus, so it's free in there. It actually comes in little rings instead of the classic DNA helix that you probably heard of and read about in books. So those are some of the characteristics. The next time that we meet, you can go straight to the microbiology set and watch those and actually go through animals and plants and do some new experiments and a couple of the old same ones, and then bacteria and also fungus. So you look at things like yeast and mushroom spores under the microscope. So it's been a lot of fun, and we'll see you in the next set of modules.